Hello, welcome. I hope everyone can see me and hear me all right. Thanks so much for joining us. This is our first Emerald Chapter program of Fall through Spring and our first live program broadcasted via YouTube. My name is Emmeline. I'm the president of the Emerald Chapter of the Native Plant Society of Oregon. We're excited to be able to offer these programs live. Please use the YouTube chat video feature to say hi. Let us know where you're watching from. And if you have any questions for the presenter during the presentation, please let us know on the YouTube chat. If you exit the full screen, if you're watching a video and you exit the full screen, you should see that chat feature below the minimize screen on YouTube. So I'm excited that the Emerald Chapter is starting to offer more events. Again, unfortunately, like so many other things, we had a lot of cancellations this spring and fall, or spring and summer. But We're shortly going to have our first ever plant lover ramble in the Eugene area on Sunday, October 18th between 3 and 5 p.m. This is going to be a casual plant meetup to be held in a local wild space, rain or shine, year round. All levels of botany and native plant expertise are welcome. This isn't a traditional plant walk led by an expert, but it's going to be a group adventure. Come ready to ask questions, come ready to share knowledge of whatever plants we find. This walk will be a short walk on a flat surface. Please pre-register by emailing your name and contact number to m_president@mpsorgan.org. We ask that everyone wear a mask, social distance along the trail, and attend only if they're free of symptoms. This is going to be a safe way and hopefully an exciting way for us to meet up, get outside together, and see some plants. Next month's program, our October program, is co-sponsored with Cascade Mycological Society. It'll be Wednesday, October 21st at 7 p.m. Andy McKinnon will be presenting two of my favorite kingdoms, plant and fungi. Our November program will be Monday, November 16th at 7 p.m. It'll be 40 years of plant walks in Alton Baker Park, the plants, the people, and climate change. It'll be presented by Biddy Roy, David Wagner, and Sarah Ward. David Wagner and Biddy Roy led plant walks in Alton Baker Park for 15 weeks each spring for 40 years. On these walks, they taught plant identification, they told plant stories, and recorded when each species they first saw came into flower. Because they were in the park over a period of more than 40 years, their records included first sightings of new invasive species. So this presentation will have a virtual tour of Alton Baker Park, tell some of the stories about the plants and people on the walks, and they'll show you how their long-term data set is useful for assessing climate change. I also want to welcome to our board Bob Hladky. He joined myself, Stephen, Margaret, and Michael this year as our secretary. So welcome, Bob. And the Emerald chapter is looking for a newsletter editor. If you have particular skills in this area, please email me at m_president@mpsorgan.org. So now for tonight's events, we're excited to welcome Dr. Tom Kaye. He's the founder and executive director of the Institute for Applied Ecology, a nonprofit organization with a mission to conserve native habitat and species through research, restoration, and education. Tom serves on the board of directors of the Society of Ecological Restoration, and he's a Courtesy Associate Professor at the Department of Botany and Plant Pathology at Oregon State University. Tom conducts research on rare species reintroductions, habitat restoration, plant invasions, and plant population responses to climate change, and he engages prison inmates in conservation through the Sagebush in Prisons project. Sourcing native plants for restoration is a key area of interest, research, and publication for Dr. Kaye. He serves as a commission member on the IUCN SSC Seed Conservation Specialist Group. So welcome, Tom. Thanks so much for joining us here tonight on this first live program of our year.
Thank you very much, Emmeline, and it's a pleasure to be here. I really appreciate everybody coming out this evening, coming out virtually to see this talk, and hopefully I can tell you something about a plant you either know well or our first meeting that might be interesting for you. And again, I appreciate that introduction, and I'm Tom Kaye, and I'm the executive director of the Institute for Applied Ecology, where I've been working for about 20 years. And I'm going to give you a talk today about reintroduction of golden paintbrush, Castilleja levisecta, to Oregon. So I'm going to share my screen with you and get started on that. So one moment while we get that fired up. And this plant, golden paintbrush, is really very showy and kind of a poster child for recovery of endangered species. There's been a lot of work done on it, a lot of restoration, and I'm really looking forward to telling you some about that. First off, the Institute for Applied Ecology, where I work, is a nonprofit organization based in Corvallis, Oregon. And our mission is to conserve native species and habitats through restoration, research, and education. And we have programs in each of those areas. And we also employ a fair number of people here in Oregon as well as in New Mexico, where we have a satellite office. Before I get going, I do need to thank a few people who have worked with me over the years on Golden Paintbrush and our funder, the U.S. Fish and Wildlife Service. So graduate students like Beth Lawrence, Katie Jones, Ian Finkston, Caitlin Lawrence, and Isaac Sandlin have all contributed tremendously to the story I'm going to tell you today. Also, various other members of the Institute for Applied Ecology staff and volunteers have participated in this work, everything from getting plants in the ground to taking data and doing analyses, etc. The Natural Resource Conservation Services Corvallis Plant Material Center has grown a bunch of the plants and the seeds that we've used. And then there's various sites that have hosted the outplantings. And so we want to thank all of those organizations and agencies that have enabled the planting that has resulted in the research as well as the recovery successes I'm going to tell you about. So golden paintbrush in Latin is Castilleja levisecta, and it is this interesting member of the paintbrush group that's bright yellow instead of red, like many species are. And they sometimes look like little golden candles out in grasslands and prairies. Really a stunning plant. And I'm gonna tell you this story kind of backwards and in flashback. This photograph here is at the Finley Wildlife Refuge near Corvallis, between Corvallis and Eugene. And this is a restored prairie that contains golden paintbrush that was seeded into it. And you can see here acres and acres of land that have golden paintbrush growing in it. There are over 100,000 golden paintbrush growing in this one population. At one point, there were over 200,000. This is the largest population in Oregon, possibly in the world, at least for a time. And so many of our reintroductions have been quite successful. This is the most successful, but others like this one have maybe not quite as many plants, but certainly the density that is spectacular and puts on quite a show in the late spring and early summer. The best time to see golden paintbrush is usually about May. It usually starts in April and ends in June. Quite a show. Historically, though, golden paintbrush occurred in populations in Oregon, Washington, and British Columbia. And you can see here, I'm going to set up my pointer, that this blue line represents the populations of wild populations in the entire range of the species. And that population size has held fairly steady or declined somewhat over the last 40 years. But since 2010, when when reintroductions began, you can see that the overall population, because of these introductions, has skyrocketed. This is kind of like the human population meets the Industrial Revolution. It's just this incredible spike in numbers into the hundreds and hundreds of thousands of plants. And most of that is all due to reintroductions, both in Oregon and Washington. And now some of that's taking hold in British Columbia as well. If you look at just the Oregon numbers, you can see that we've gone from no plants, none in the wild in Oregon, up to hundreds of thousands of plants of our own in 2018. And in 2019, we saw a little decline, but the population's still very substantial, very large. And this has been the result of outplanting primarily of seeds. In this case, about 25 pounds of seeds over the last decade. Maybe that doesn't sound like a lot of seeds, but that's been the total output from the fields that we've grown seeds in. And but that equals over 57 million seeds. So while we may be talking about hundreds of thousands of plants, they're the result of many millions and millions of seeds. And that's why we've been able to get these big numbers of plants, is that the the seed we can put on the ground has been so copious. If you look at not just the total number of plants, but the number of populations, which might actually be a better way to look at it, you can see, though, that we've grown from no populations in 2010 up to about 23 populations in 2018, and we've held there for the last couple of years. The populations that are over 1,000 plants, though, those are the populations that count towards the U.S. Fish and Wildlife Service's metrics for recovering the species and delisting it, taking it off the endangered species list. That number of populations has also crept upward, and we're in the neighborhood of 8 or 9 populations that meet that single criterion. So we've got some major successes from total absence from Oregon up to this. But how did we get there? How did this happen? Well, this story I'm telling you is really told in flashback. And so going back a ways, Thomas Howell was the first botanist, a white Euro-American, who described the species for science. He collected it, and it was described by Bradshaw. And this species was new to him, was new to everybody who saw it. And it occurred where he collected it in a place called Mill Plain near Vancouver, Washington. And that's the site of Fort Vancouver. And that's probably why Thomas Howell was there. He probably went across and saw Fort Vancouver. Now, you know, this was in the late 1800s, and there wasn't a whole lot of a population around in the Mill Plain area at the time. This image is from even earlier than that, but you can see there's a broad plain out back behind Fort Vancouver, looking upriver. And what that looks like today, however, is city. This is the city of Vancouver. You know, you've got major highway, I-5 running north-south. You've got major train tracks. You've got neighborhoods and surface streets, and the habitat's essentially gone. There's a few places in the Mill Plain area where there's some open fields, but largely the habitat for prairies and prairie plants has been obliterated. Where that habitat remains and where the species currently remains in wild state is largely in Western Washington. And here's a picture of Mima prairie. This is a kind of landform that occurs in Western Washington, just south of the glaciated Puget Sound. And it's a mounded prairie. It's got all these interesting bumps all over it. And the golden paintbrush largely occurred on the hills and not so much in between them. This is an image looking from much higher up. There's a lot of controversy over how these kinds of mounds were formed in the first place. Some folks think that it's due to pocket gophers that were very abundant and formed these big mounds where they had long-term colonies, and the gophers were making the mounds themselves by bringing up soil and mounding it up. Others think that this is a periglacial phenomenon. This is something that occurs on the margins of glaciated areas, largely through the action of freezing and thawing and erosion from overland flow of water. The problem is that explanation can't work for all parts of the world where these kinds of mounded prairies occur. Some of them are in areas that have never seen glaciation. But in this context, it looks like it's possible there are more than one mechanism for the mounding process to occur. And I ascribe to the periglacial phenomenon, patterned ground school of thought, but who knows? It's fun to speculate. Back in 1984, to continue this flashback, I was a college student at the Evergreen State College, and this is a very grainy picture of me back at that time. And you can see I'm wearing a hat that has a Nature Conservancy patch on it. And I had a weekend job working for the Nature Conservancy, going out to a prairie nearby called Rocky Prairie, that was another mounded prairie, and being a presence because there was a problem. This prairie that was a preserve was having people drive vehicles over it. Off-road vehicles were coming just to do donuts and have a good time. It was a lot of fun to drive over those mounds and things. But the prairie was so sensitive and so full of diversity and rare species that they really wanted to deter that. So putting up a fence wasn't enough, putting up signs wasn't enough, but maybe stationing a person there just to be a presence and a deterrent would help. And so that was my job for several weekends in a row in the spring, was just to go hang out at this prairie. And that's how I met this plant, this golden paintbrush, because this is one of the places where the plant still occurred. And it was a wonderful experience. And I got to know this plant just by hanging out with it. And over time, I lived in Washington a little longer, and then I moved back to Oregon. And it became one of my interests to see if I could find this plant in Oregon again, or maybe help bring it back. And so flashing back again to 1938, this is a plant specimen from a herbarium. And this is the last specimen of golden paintbrush that was collected in Oregon. It was perhaps seen by people in the area where this was collected. It was collected in a place called Peterson Butte Cemetery. This is directly east across the Willamette Valley from Corvallis. And some of the folks in that neighborhood who I've corresponded with say, I remember that from when I was a kid. And it may have persisted up until the 1960s or early 70s, but it's definitely gone now. I went around and looked. And so one of the places I went was Peterson Butte Cemetery. And this is what it looks like today. It's a cemetery, and you can see the butte behind through the trees. And there's no golden paintbrush growing there anymore. It's gone. The first time I went there, I did find some other rare prairie remnant plants, but even those seem to be gone now. It's a well-tended cemetery. So now more about golden paintbrush. The species was listed as threatened in 1997. And at about that time, these little green dots were present. These were the remaining populations in the wild. So there was one in southwestern Washington called Rocky Prairie, as I mentioned, and there's some up on Whidbey Island, and there were some up in the San Juan Islands and on Vancouver Island. There are now a couple of populations that are on islets or small islands just off the shore of Vancouver Island outside of Victoria. So there's still some populations there. It's a plant in the Scrophulariaceae, or more recently, the Orobuncaceae. It's a grassland plant only, prairie plant. It's endemic to the Pacific Northwest. It doesn't occur anywhere else in the world. It's a perennial, and perhaps the most interesting part of its biology is that it's a hemiparasite. In other words, it's a parasitic plant, but it's chlorophyllous too. It contains its own chlorophyll and can photosynthesize. So it's only a hemiparasite because it can live on its own. As a parasite, it can connect to other plants. And some of the plants it can connect to are this plant down here, which is Romer's fescue on the left. And this one over here on the right is woolly sunflower, also known as Areophilum linatum. And these two plants are both very common in prairie remnants and were almost certainly important historical parasite hosts for golden paintbrush. And one of the things that we wanted to know early on in my research on this plant, before we started reintroductions in Oregon, was more about the mating system of the plant. How did it breed? Is it susceptible to inbreeding, or can it be a selfer? Or is it susceptible to outbreeding depression, or is outbreeding good for it? We knew nothing about the species' basic reproductive biology. So this was a chance to start from a position of some knowledge about it. So one of the things we did was some crossing experiments. And these are the cross types we experimented with. On the left here, we have selfing, where we took pollen from a flower on a plant and put it on another flower on the same plant. So it's self-pollen. Another thing we did was a cross to a sibling. So this is close to selfing, or it's certainly inbreeding, to put the pollen on a close relative of the plant. And we knew they were close relatives because we grew seeds from a single capsule on a maternal plant, and we grew those out. And so every plant from that capsule had to be at least a half sibling. They had the same mom and perhaps a full sibling with the same dad, but we couldn't be sure about that. So when we crossed to siblings, we also crossed to other plants in the same population. Just this would be what a pollinator would usually do, just cross to plants around. And then finally, we crossed to plants from different populations. So these were the kind of crosses that normally wouldn't occur now with fragmented habitat, but could these crosses work? We wanted to know. Over on the left is inbreeding, and we're looking for inbreeding depression in particular. And over on the right is outbreeding, which actually can result in outbreeding depression, which is a loss of fitness compared to the parents, or it can result in hybrid vigor, a heterosis, an improvement in fitness. So how did we do this? Well, here's a flower of the golden paintbrush here, and we took the pollen here from the stamen and moved it up here to the stigma. Another way to see it is here is on the plant. We peeled back one of the bracts on the outside that are so showy in paintbrushes and found a flower inside. This is actually the corolla down here. Oh no, sorry, these are the sepals, and this is the corolla right here. Right here is the corolla tube coming up here. And this is where the pollen is contained, right here, near the top of the flowers where the anthers are. And this is the stigma, where we move the pollen to. I had some help with this from a variety of interns that were learning about conservation biology. So they were helping move these chunks of pollen to the stigmas. And we actually used different colors of thread to mark the flowers and therefore the developing fruits so that we could distinguish between them in the treatments we had applied. And here's some more showing the thread tied around the base of a fruit. So as the flower develops a fruit, that thread stayed there. And this is what we found, that selfing actually resulted in very poor seed set. This is just terrible seed set. It's almost zero. Crossing to a sibling did give us about 35% seed set of all the ovules that could make a seed. Only 35% did make a seed. If we crossed to the same population, we got about 70%. And if we crossed to another population, we got almost 80%. So we found that the plants are not capable of autogamy or selfing, and also they show strong signs of inbreeding depression. But when we cross to another plant in the same population, that becomes a baseline for this measure. And comparing crosses to another population showed that we actually had an improvement in fitness when the recipient of the pollen was from another population. So distance was actually good for this, possibly because of the bottlenecking or inbreeding depression in existing populations. Now, the same pattern held up when we looked at seed germination and at the growth of the plants that came out of these seeds in this figure. So this wasn't just seed viability or seed set, it was all aspects of plant growth after that. Another thing we've learned since then is that the pollinators of this plant, which it cannot survive without, are bumblebees. And not only that, but in most populations in Oregon and Washington, the predominant pollinator is Bombus californicus, the California bumblebee. One species of bumblebee is responsible for most of the pollinations. And that creates a vulnerability for this species, so that if this species loses its pollinator, and we know that bumblebees are in trouble, then this species will be in dire straits. Another flashback, Morton and Jessie Peck. Now, Morton Peck and his wife were intrepid botanists, just like Thomas Howell, and they went on horseback into the boonies. They went all over Oregon collecting plants. And Morton Peck wrote the first flora of Oregon, and he worked at Willamette University in Salem, but he took all of his trips from there. And he he collected a specimen of, actually a few specimens of golden paintbrush in the Salem area. And on his herbarium label, he wrote that the plant occurred in moist, open ground. Well, one of the things we're questioning here is, what kind of habitat did golden paintbrush grow in Oregon? I mean, there weren't any populations left. It was completely extinct. And the labels we had were generally vague on exactly where the plants were. The one from Peterson Butte Cemetery was by far the most specific of locations. And that's an upland prairie with gravel and gravelly soils. But when you say moist open ground in the Willamette Valley, that kind of suggests wetland, because we have a lot of wetland in the bottomland around Salem. So one of the things we tried to do was plant this species in both upland and wetland conditions, because we wanted to know if it grew in wetlands, because if that was true, it opened up a whole lot more habitat where we could put this species. And so we planted golden paintbrush in multiple populations, 9 locations in Oregon and 1 location in Washington. And some of these were wetlands or partly wetlands, and some were uplands. And so we planted it all over these places. And then we came back the next year to see what happened. This is an example of an upland prairie, and this is an example of a wetland prairie, both occurring at the Basket Slough National Wildlife Refuge. And what we found was, in a very simple graph, that plants in uplands did pretty well and plants in wetlands did not. And although there was an indication that the plant occurred in moist open ground near Salem, it's pretty clear that wasn't wetland habitat. Because all of those plants perished in just a year. And another experiment we did was to grow plants in similar kinds of situation, but we wanted to know which populations actually that occurred in the wild still grew well in Oregon. Because the plant was completely extirpated from our state, we had to collect seeds for reintroduction work here from somewhere else. And we needed to know what's the best place to do that. The closest place was that place I mentioned earlier called Rocky Prairie, but there are many other populations up here in the Whidbey Island area and on Trial Island and San Juan Island. And we wanted to know if we could use those and screen them for which ones worked best. But in the process, and so we grew them together in what you call a common garden. And in the process, though, we could measure things like, how far is it from where we got the seed to where we planted the seed? And so that was geographic distance. We could also measure how similar the habitat was where we collected the seeds to where we planted them on the ground. And so we're looking at both geographic distance and environmental or ecological distance. So what we found here, and this was from my student Beth Lawrence, is I'm going to focus here on the right-hand graph here. And what this shows is that as the community distance, the ecological distance between a source population and where we planted them in Oregon, as that distance increased, we saw a decline in plant survival. In other words, when we planted them in places that were much like where we got the seeds, the plants did better. They did better in habitat like where they came from. But they did poorly when the plant community was more different from where they came from. This didn't apply so much to soils as to the plants themselves. It also didn't apply to the geographic distance. As geographic distance increased, there was essentially no difference in how well plants did. The difference that mattered was how similar the plant community was. And over here, what we're showing is an ordination, and these kinds of symbols in open triangles are what the sites where we planted them look like ecologically. And over here is what the sites where we collected the seeds looked like ecologically. And then these are sites where the plants tended to do quite well because they were more like the source populations. Now, one of the things we needed to do in order to scale up and do large-scale reintroduction in Oregon was to get seeds of this plant going. But in order to do that, we needed to understand more about the hosts of this plant. So what I'm going to tell you right now is how this parasitic plant interacts with other plants. And here's its parasitic apparatus. These items right here are called haustoria, and they're what a parasitic underground plant uses to connect to the roots of another plant. So these sort of ball-shaped things are on the tips of roots of golden paintbrush, and they've attached themselves to the root of a yarrow plant, Achillea millefolium. And so they're literally attached to the root of this other plant, and they're pulling resources out. They're taking fluids like water and fluids that contain sugars and other secondary compounds that might even be confer a defense to this plant. And so here's some of the things we found. That without a host, it grows okay in a greenhouse. With a host of woolly sunflower, it grows okay, about the same. When you grow it with a grass like Romer's fescue, it actually grows not quite as well as if it was alone. But if you grow it with both, it grows even better. So this is a suggestion that the plant has a varied diet. This is when you grow it alone, and this is when you grow it in a mix. Well, that's pretty cool. It likes a varied diet. But when we took these same plants and we planted them out in the wild and waited a year, here's what we found, is that without a host, the plants did fine. They did the best, actually. With the grass, they actually did almost as well. And with woolly sunflower, they did worse. This is the opposite of what we found in the greenhouse. And with a mix, they did, pretty well, but still worse than when we grew them alone. But there are other plants growing out there. And so these plants that were grown alone or grown alone and then planted in the wild had a chance to connect to other plants. But why this? Why this opposite pattern? Well, here's what we found. Voles. Voles were the explanation. The thing with voles is they like to tunnel in the ground, and they're eating the roots of other plants, and they're eating the plants above ground as well. And one of the things they like to tunnel under is woolly sunflower. They appear to like the taste of woolly sunflower and eating the roots of this plant. And so without a host, we had very few tunnels. And with the grass, Romer's fescue, we only had a few tunnels. But where we planted woolly sunflower or a mix, which means woolly sunflower was there, we had many tunnels. And the tunnels were lethal. So the answer to the question of why did we see this opposite is because in the wild, we saw lots of voles, and the voles were eating the woolly sunflower. And the problem with that was that it was an odd year for the voles. Voles have up years and down years, up years and down years. And this was a big up year for the voles. And it just so happened that we did this experiment on a year with many, many voles. Most years are not like this. And usually woolly sunflower can be a good host, even in the wild, but not when there's an outbreak of voles. So it's not just woolly sunflower and Romer's fescue that can be hosts. That's what's shown here in the upper two left-hand species. But we also looked at other species by growing golden paintbrush in a greenhouse with other host plants, like those two, or this is the prairie June grass, Castilleja cristata. This is self-heal, Prunella vulgaris. Over here is the yarrow, Achillea millefolium, or the rose checker mallow, Castilleja virgata, and more. We planted it in the greenhouse with these things, and then we looked at the roots. And these, all the plants on the top have roots with haustoria coming to them. These are all grasses. All of them are grasses. And then in the next row, we have two in the composite family, a plant in the mint family, in the mallow family, another one in the verbena family. So this parasitic plant appears to be a generalist. It does not seem to prefer any particular host just to make an attachment to. It'll even attach its roots to itself, which is kind of a new kind of plant disgusting. So it's a generalist. It'll parasitize anything it runs into underground, but that doesn't mean everything is an equally good host. Just because it can connect to the roots of another plant doesn't mean that other plant is necessarily good. It just means the golden paintbrush isn't very discriminating. So we wanted to find out, does the neighbor identity matter when these things are growing out in the wild? And we already saw that that can be complicated by voles, but we tried this on a much larger scale with a lot more kinds of species. And so one of the sites we worked at was right here at the Willamette Valley National Wildlife Refuge, again, between Corvallis and Eugene. And we planted the plants out in this grassland. Also, we picked another piece of grassland next to it, and we used herbicide to kill all the standing vegetation. We used Roundup or glyphosate to do so. And that meant that when we plant a golden paintbrush in the ground and we planted it with a host, that's the only host it has. When we grow it out in a wide field, it has access to other hosts that happen to be there. So I'm going to tell you just about the results from this experiment where we planted with single hosts, and I'm going to ignore for the time being this multiple hosts or ambient vegetation opportunity. And what we found was that the predicted probability of survival of these plants statistically was actually higher when we grew them with grasses generally than when we grew them with forbs. Forbs meaning flowering plants. So the host species did matter, but in kind of general ways. Grasses were good for survival. But when we looked at the probability of flowering, grasses weren't actually any better than planting alone, but planting with forbs, at least two of them, yarrow and woolly sunflower, resulted in higher flowering than plants grown without a host out in the wild. But some of these hosts, like Prunella vulgaris, the self-heal, are actually much worse to grow with than even growing it alone. So not all hosts are the same. They actually affect the plant in different ways depending on the life history component you're looking at. And some hosts are good and some hosts are bad. To look at that difference between hosts in a different way, I want to call your attention to these graphs here. What these show is the survival of plants in relation to the richness or the diversity of other species growing around the perimeter of the plant. These were measured within 10 centimeters of the rooting position of a transplanted golden paintbrush. And what we found was if we looked at perennial plants only, we found a positive relationship. The more different kinds of neighbors a plant has, the higher its survival from one year to the next. So this is reinforcing that varied diet concept, that the more kinds of neighbors a golden paintbrush has, the more likely it is to survive from one year to the next. So species diversity, species richness around these plants really matters. So when we're looking at planting into prairies and restored prairies, we need to be thinking about finding places with high species diversity because that affects plant fitness directly. But now I'm going to call your attention over here to annuals. What was going on if we planted in a place with lots of different kinds of annuals growing? What we found was the opposite. So the more kinds of annuals that were growing around a golden paintbrush resulted in fewer golden paintbrush surviving to the next year. Now, why would that be? Why are perennials good and annuals bad? Well, if you think about what annuals do, what are annuals good at? Well, after they flower, they die. That is what an annual does. And if you're a parasitic plant and you've spent some of your currency to establish a connection to this annual plant and it dies, that's not as good as if you'd established a connection to a perennial plant that's going to live through the winter and into the next growing season. So it's really a matter of throwing good money after bad. Now, I mentioned that we needed to produce a lot of seeds to get into the restoration business for this species. So one of the things we did was work with the Corvallis Plant Materials Center of the Natural Resource Conservation Service to grow golden paintbrush in an agricultural context, to just get lots and lots of seeds. We want pounds and pounds of seeds so that we can get these out on the landscape. And so we looked at the best sources, the best sources to put into production. And those were from that research we did earlier. Remember, we planted plants in common gardens from lots of different locations. Well, we picked the top 3 populations they were actually from Washington, mostly from the Whidbey Island area. And we also threw in one more, which was the population at Rocky Prairie. And we threw in like 12% of that. And it's one of those cases of not blindly following science, because the Whidbey Island plants are from an area that's more maritime. Although they did grow here very well, they may miss something that we need here in the long term. And even though Rocky Prairie plants were actually, even though they're geographically closest to the Willamette Valley, were the ones that grew here the worst, we put them into our agricultural field mix anyway, because we wanted some representation of those genes, because they're from fairly similar kinds of prairies. So another reason we put a mixture of different populations together here was because we wanted to leverage that heterosis, that hybrid vigor we got when we crossed between different populations in that mating system study, so that these seeds we got out of here would have a fitness boost when we first put them out into a new site. And so, what we found is that both transplanting and seeding directly can work. These plants on the right are all transplanted into a prairie. This is my colleague in Washington, Peter Dunwitty, who's done very similar and spectacular successful work in golden paintbrush reintroduction in those prairies. And here's a transplant in Oregon that's doing fine. This, however, is a field where we sowed seeds, and we can get thousands, tens of thousands, if not just hundreds, from one application of seeds into a restored prairie. Whereas for plugs, it takes a lot of work. We've got to grow them in the greenhouse, then bring them out and plant them individually. It's a lot of labor compared to walking around outside for a few minutes and scattering seeds. So there's a attractiveness of using seeds, and it tends to work pretty well, at least some of the time. Transplants survive to a proportion of about 50% after the first year. Overall, there's a lot of variability to that. And seeds come up as seedlings or golden paintbrush plants to the tune of about 0.2%. That sounds terrible, but if you've got 1 million seeds, you can overwhelm those odds and get populations to establish. And that's the math that we've leveraged to make this work. Now, one of the questions that we have faced is, should we really use a mix of populations to establish a population in Oregon, or should we use a single population? And one of the questions we have is, when we do use seeds that are from a nursery that is established from a mix of different populations, does that genetic diversity actually carry through to the reintroduction sites, or are we just fooling ourselves? Because when you get into a domesticated environment, you can get very imbalanced seed production from different population sources. And so we wanted to check to see if that was the case. And we worked with a graduate student at Northwestern University to examine this. And so Adrienne St. Clair did this research. And what she found was that the wild populations each had a different genetic signature, and we could track that genetic signature through these different contexts. Now I'm just going to look at this example that's going into Oregon. So following these arrows down, we get to the Oregon nursery. And then we find that in the nursery, the different rows of plants we planted, which represented different populations, held true generally. Those populations genetically looked just like they did in the wild. So that's great. We've got the representation genetically in the nursery that we think we should based on where we got the seeds. But then what happens when we put these out in the wild? And it turns out that we get this mix of genetics that resembles the mix that we offered from the nursery. So the genetic diversity that we're creating in the nursery is actually holding forth in the F1 crosses between the different lines that are in the nursery and showing up in the wild. So that's what we wanted to find out. And what we learned is what we think we're doing is actually what's working. And so we're very pleased about that because it could have gone another way. Moving on, this is where we are today with populations in Oregon, in the Willamette Valley. This shaded area is the Willamette Valley ecoregion, and we have upwards of 23 populations overall that have been established in this region. Many of them are large. Howell Savanna is a very large population. Let's see, the Finley Wildlife Refuge has very large populations, and so on. Some of them are smaller that haven't done as well. And so when we started reintroduction like this, sometimes it's kind of like buying a lottery ticket. Sometimes it works really well and sometimes it doesn't. And you just have to keep buying more tickets if you want to win more often. But you'll notice that in the introduction or the title of this talk, one of the things I said was that is this a success? And basically my title was a question mark. Has reintroduction in Oregon actually succeeded? And when we look at some of our introductions, we find a story that is cause for concern. For example, we see a boom and bust cycle, especially where we plant seeds. You can see over time, we get, after 2 years, we get a bounce in the number of plants, but then a decline. And that happens multiple times in these examples that I'm showing you here. That's cause for concern. The hope is that these plants kind of reach an equilibrium and then stabilize, and that it's okay that we have a boom and a bust as long as they stabilize at some reasonable population size. We also see something similar in most of the plugged populations and different techniques, but the plugs also tend to die and fade out over and over again. We can see that happening. But oftentimes, or sometimes, the plugs do stabilize a little bit. They have their ups and downs and stabilize. We have seen some of that happen in plugs, but we've seen some populations decline. In Washington state, the plugged populations, those have been established just by plugs, have all faded out almost entirely. It's not been a successful technique in Washington. But we also see the same boom and bust cycle in Washington from seeds. So we have some concern that these populations are viable, that have a long-term sustainability. And if they don't, then our initial glee and optimism at seeing large populations established is tempered and perhaps defeated. So this is one of our most important questions now, is what happens to these populations long-term once we take our foot off the pedal and we stop adding more and more seeds repeatedly to the same sites? What do they do? What do they do? Do they coast out and fade away, or do they reach an equilibrium? And how do we manage for those plants in those prairies? What makes them tick? We don't yet have a lot of information about how plants respond to different prairie management techniques. We think they respond very well to fire, but mowing also seems to help, certainly to knock back thick grass and woody plant invasion. But what about some other techniques? We need to dig into that and get some answers before, at least I'm very comfortable saying this species is recovered. Fire, shown here, is a good technique for not just managing the populations we think, but certainly for creating a seed bed in which to put seeds and establish a new population. I'm going to show you a few pictures of just some other restored prairies in the Willamette Valley. Restoration, actually, of the whole prairie ecosystem can work. And it's that ecosystem, that entire plant community, that we need to restore in order to keep golden paintbrush and bring on bigger populations. Because one of the things we know is that it needs a lot of different kinds of neighbors. It needs a diverse ecosystem. This is one of the superpowers of biodiversity, is supporting other species like golden paintbrush. This is at Ankeny Wildlife Refuge. This is a restored prairie shown in the foreground. Here is this pale purple to white colored flower called Willamette daisy, or Erigeron decumbens. This is completely restored, and the Willamette daisies seeded into this site. This is now the second largest population of Willamette daisy in the world, and it's restored. There's also golden paintbrush here, and it's doing quite well. Here's golden paintbrush in a prairie near Eugene. This is out near the Fern Ridge Reservoir. And one of the interesting things about this population is that the golden paintbrush is parasitizing woolly sunflower in the background here, but it's also parasitizing this lupin, which is Kincaid's lupin, which is also the host plant for the Fender's blue butterfly. So this is a suggestion that as we restore prairies and we put more and more of these species into them, that, say, if we put in Kincaid's lupin, we're supporting golden paintbrush as well, and the Fender's blue butterfly. So when we restore diversity, we actually get this positive cascade towards supporting more species. Now, for the last several years, we've been counting golden paintbrush, and we've been using support from interns like these guys here that are studying plant ecology and conservation biology. And these interns are supported by grants from the Native Plant Society of Oregon. So we thank NPSO for the support of these interns who are going on to more jobs in conservation and are learning quite a bit about plant conservation. And these guys helped me a couple of years ago with our range-wide survey of golden paintbrush in Oregon. So they became very adept at sampling populations for this species and getting our counts up to date, because we won't know if this is working unless we are able to count these plants every year. And currently, we're without funding to do conservation monitoring for this species. We're at the end of our cycle with funding from the U.S. Fish and Wildlife Service. Here's some more interns gathering data. And I'd like to close with a comment about conservation of species that help each other. So this butterfly here is the Taylor's checker spot butterfly. This species has a few populations in Washington and only 2 populations left in Oregon. There is an attempt to captively rear and introduce this species back to more sites in Oregon. And some of those have golden paintbrush. Why would that be? Well, it turns out that the host plants for this butterfly historically was probably predominantly this paintbrush here, the harsh Indian paintbrush, but also golden paintbrush. Remember I mentioned Rocky Prairie? Rocky Prairie is a historic site for Taylor's checker spot and is still a site for golden paintbrush. So we know the butterfly used to use this paintbrush as well. Interestingly, the butterfly has made a host switch in Oregon entirely to English plantain, a weedy plant. And it's only because the English plantain still has invaded and now occurs in some of these prairies, while the paintbrushes have faded out, that we still have Taylor's checker spot. Taylor's checker spot would be extinct in Oregon if it weren't for a weed. So that's an interesting twist on conservation. But we're trying to bring back more diversity to the populations of Taylor's checker spot. And one way to do that is to add golden paintbrush back into the mix. There has been some controversy over whether Taylor's checker spot actually can use the golden paintbrush, and it's very clear that it can. This is a caterpillar that is using the golden paintbrush in winter. So that was a unknown thing. We didn't know that it could actually use golden paintbrush in the winter. Here's a caterpillar that's also using the English plantain. And here's English plantain over here too. And here's an egg mass on an English plantain. So we know they like English plantain. They do very well on it. They perhaps even prefer it at this point. But they can also do well with the paintbrushes, and adding diversity actually creates a bet hedging strategy in case the plantain has a bad year. This is what happens when you add golden paintbrush into an existing population of Taylor's checker spot where the paintbrushes had gone out. They recolonize on the golden paintbrush. And so you see these caterpillars just munching away on the golden paintbrush. You might say this is contrary to golden paintbrush survival, but it's actually, I think this is an acceptable use as long as we can get the populations up there. So this is just an example, I'm going to close here, of how working with a federally threatened species like golden paintbrush can actually cascade into more conservation advantages to species like the endangered Taylor's checker spot, and working in other species like Kincaid's lupin then cascades into helping butterflies like the Fender's blue butterfly. And in this manner, where we have very limited numbers of places where we can do prairie conservation, we can actually stack our prairie conservation priorities in the same site and still have wins, instead of having these species be at odds with one another or only at one place or another. We can bring them together and leverage what limited space we have. And given the time, I'm going to stop there and turn it over to questions, and I hope this has been interesting for you. So I'm hoping Stephen or Emmeline can come on. Well, until they do, I'll keep talking. Here's another twist on this. This plant here, the harsh paintbrush, is a plant that comes in two different flavors, a diploid and a polyploid, one set of chromosomes or multiple sets of chromosomes. And it turns out when you plant the diploid form next to golden paintbrush, they form a hybrid swarm. But what we wanted to find out was what happens when you do these crosses between polyploids. And this is a graduate student of mine, Ian Finkston. I'm sorry, Isaac Sandlin, sorry, Isaac, who did these crosses. And what we found was that when we cross diploid to diploid, we get very free hybridization. But when we cross diploid to tetraploid or hexaploid, which are the polyploids, we get very low hybrid vigor. And so we see that we can plant them together and avoid hybrids if we plant the polyploid race with the golden paintbrush. If we plant the diploid race, we'll get a hybrid swarm, and we'll start breaking down our golden paintbrush genetic integrity, and we'll lose populations that are useful for recovery of the species. And that has occurred in Washington, where the plants were planted together. But we have a loophole, the polyploid loophole, where we can plant the polyploids and get away with it, and we can have all of them growing together. So now I'll stop and let Emmeline in.
Okay, thanks, Tom. I didn't want to interrupt your twist at the end there.
Thanks.
Thanks so much for joining us, Tom. I'm going to turn it over to Stephen, who hopefully can hop on and hopefully has some questions.
All right. Thank you. I think Stephen's going to manage the chat stream and synthesize questions for me.
All right, I'm here.
There's Stephen, good.
Yeah,
Thanks.
I want to thank Tom for presenting this evening. That was very informative and a wonderful presentation. This is our second Emerald Chapter virtual presentation, but our first on YouTube Live, and I would say this is quite a success. We do have a couple questions, Tom. Our first question is, have you assessed the role of herbivores in the decline of populations?
Good question. What's the role of herbivores? Well, certainly we have found in all these experiments we've done that some herbivores like voles can really take down plants. but deer can as well. And so where there are significant populations of deer, we've seen pretty significant herbivory. And that's actually one of the problems in some of the wild populations in Washington that remain as well. Some of those small and few wild populations that get grazed, basically they have reproductive failure because they're not making seeds anymore. And some of those populations appear to be being driven down directly by herbivory from deer. In 1984, when Beth Lawrence was doing her common gardens, we started putting out plants and the deer seemed to come right to them. So she turned to and built deer fences around all 10 of her common gardens, which was a huge task. You never met a woman who could do more. I mean, Beth was incredible. And that helped, that saved the experiment. But it also pointed out that that could be a liability in the future. Generally, we don't see huge amounts of herbivory from deer in any given year, but it can happen. It's the same thing with voles. We don't usually see a lot of it, but in some years that are peak years, it's a problem and they do kill plants. What's the next one, Stephen? Stephen, we're waiting for you to come up with another
great.
question.
Thank you. We have one more question. Are seeds available to homeowners, or do you know if they ever will be available?
Oh, great question. Generally not. The seeds we have limited supplies, and we really try to focus those on restoration sites on public lands. One of the issues around that is that for recovery purposes, a population needs to be on protected land. So that land needs to be either public or in a conservation easement. And so because these seeds are grown with money from the US Fish and Wildlife Service, whose goal is to see these plants contribute to recovery, we generally don't put them on private lands without some kind of conservation overlay. There can be exceptions, and we can talk, but it's generally not for pleasure gardening. Not yet.
And then our last question here: does golden paintbrush reduce the vigor of Kincaid's lupin when it is the host?
Great question. Yeah, it is a parasite after all, and it can remove important resources from the plants. I've never seen it looking like it's damaging Kincaid's lupin, but in some of our experiments, we found that as the golden paintbrush becomes larger, the host plant loses stature and becomes smaller. And so some of the plants like yarrow or woolly sunflower can be susceptible to that. So I think depending on the context and how much of a varied diet the parasite has, that it actually can reduce the stature of some of the hosts, but maybe not very much. There is another experiment we did to look to see if we could use golden paintbrush really in a restoration context to reduce competition from dominant grasses. And we were not able to establish a high enough density of golden paintbrush to do that. However, there are other parasitic plants that have been used that way successfully in restorations in Europe. So it still could serve some of that function. We're just not sure. Well, was that it? All the questions?
I have a question if you don't mind, Tom. I'm wondering if you could let us know some good places and some good times of year to see golden paintbrush.
Sure. The easiest place to go see golden paintbrush is at the Finley National Wildlife Refuge between Corvallis and Eugene. And you can drive down the main road, and there are some populations that are right along the road, and you cannot miss them in May. They're very conspicuous. They're right up against the edge of the road. And so that's by far the easiest place. If you want to go up to, say, Basket Slough National Wildlife Refuge, you can go up there and take the trail up to the viewing platform, and you'll see paintbrush on both sides of the trail there as well. Those are all introduced. There are places in the Eugene area, but they're less accessible because they're on Army Corps lands near the Fern Ridge Reservoir, or on city property that are part of easements, like at Coyote Creek. But they are out there, and if you can get permission, you can get out there and see those as well.
Awesome. Well, I don't see any remaining questions there in the chat. So thanks so much, Tom, for joining us, and everyone who's still listening. Thanks so much for making this format a success today and this presentation a success today. I hope to see some folks at our Plant Lovers Ramble and at our upcoming programs in October and November. Take care out there. Thank you so much.
Great. Have a good time on the Ramble, and at your next meeting with Andy McKinnon, the two of my favorite kingdoms, plants and fungi. That sounds like a really good one. I'm looking forward to that myself.
All right. Thanks, Tom.
All right. Thank you, guys. Bye-bye.
Good night, everyone.
Stay well.